Good morning, class. As I was talking to you in class, last class, this chapter is a chapter based on the cell. This is your chapter 6, and I have decided to put it online, so it is available to you along with my audio explanations as a PowerPoint. And you are required to go online onto Blackboard to listen to this. If you're already opened it up and listening to it, this means that you are already onto it, which is positive, and you can listen to it and come to class prepared after you have understood it, which will help us when we would like to work on the worksheets in class. Okay, this is part of your test 2, and this is your chapter 6 based on the different aspects of the cell and different cells that we ought to know for this syllabus. Okay, as usual, my second slide always contains the most important things that you need to know about this chapter. We start with talking about just the cell. The cell, as you know, is the basic unit of life. Okay. And before the advent of the microscope, people really did not know that living things were composed of cells. They really did not know so much detail because they couldn't see it. So the pioneer amongst them was first scientist. His name we remember and revere for this process is Robert Hooke. Now Robert Hooke came up with observing and documenting the dead cells of cork. Okay. Then we have another scientist. His name is Anton von Leeuwenhoek. Now this scientist is credited to creating the first microscope that enabled people of those times to see things that were smaller in size, which the human eye couldn't really see and decipher the difference. So this gadget that he has made here is a microscope, and it contains one sample. Basically, the side here explains that this is the tip of the nail or the pin which holds the sample, and this is the lens which has been ground and polished. So from this side he can have a look and appreciate. So this is his eye here. This is the person. You can see it. Okay, you can see and appreciate material which is smaller in size. Now he is basically a Dutch glass maker, and with his construction of a microscope, he impressed the Queen of England and showed her what he called as animalcules, and impressed her enough to get funding for more work for his laboratory. Now this chart here sort of gives you the sizes in perspective. I already did a part of it in the lab when we were doing microscopy, where I had given you a chart wherein we could sort of lay gradations with respect to meter, and then centimeter, then millimeter, then micrometer, and then nanometers. Now you have cells which are individual functioning units which can vary in sizes depending on what kind of cells they are. Okay, now a chicken egg is also a cell. A nerve cell is also a cell. Nerve cell, the size could be, or the length could be close to approximately a meter or so. A chicken egg could be anything between from being about 10 to 15 or to 20 centimeters in length. Now, frog eggs range from millimeters to a centimeter. Then the cells that you and me are going to talk about in this class are ranging in the sizes from 100 micrometers to close to the bacteria, which is almost a micrometer. The smallest bacteria can be even of the size closer to the nanometer range. Now, I am guessing this gives you an idea of what are the different ranges of sizes in which cells can exist. And beyond that, what a light microscope can make it clear for you to see, and what you need an electron microscope for to see finer details. Okay. Now, the different microscopes and different microscopy techniques can enable us see any cell or any material, any sample that we want to look at, and sort of enhance certain regions and make it clearer in that fashion. Now, if you want to look at any live organism, which you will probably be doing in lab 3 on diversity, wherein you would be hopefully able to see a live organism, in which case you should not stain it, you should not dry it. That could sort of kill that organism and you really can't appreciate its difference. So that, because it is not stained, what you have to do is sort of play with the light and give it a little bit of contrast, which is what will enable you to appreciate that there is in that sample of water or colorless medium that you are able to appreciate presence of a live organism. So a bright field microscopic, unstained bright field microscopic technique is what will be helpful for you to see a live organism. Okay. Then you could also stain the specimen, which sort of enhances the contrast, and you can see, as you can see in this particular slide. You can also use different kinds of phase contrast microscopy, which amplifies the variations in density within the specimen. Again, this is not yet stained, okay? It just amplifies the variation in density or the thickness of that particular sample that you're looking at. Then there is other microscopic techniques, details, wherein you can look at the difference in density, which will make it look or appear three-dimensional. Then we move on to more sophisticated techniques of electron microscopy. Now this one, there are two kinds. Now electron microscope is not something you and me can use for this particular course. Perhaps if you are studying more in biology and in details of biology in the research field, maybe there would come a time that you would be using electron microscopic techniques to study the sample that you are looking at or you are working on. Simply put, electron microscopy does not use light as its source. Okay, it uses electrons to bombard the sample that you are looking at. Now there are two ways to look at this sample. First of all, appreciate that the sample that we are going to be looking at is extremely small in size, and you're looking at finer details, like not just the bacteria under a light microscopy, you would be able to look at it in the field just looking like this. Now imagine if you had to look at the details of this guy, see what he has inside and outside. For this, you need an electron microscope. Okay. Now, this is your light microscope, which can show you this. Now, electron microscope can be of two types: scanning electron microscope or transmission electron microscope. Scanning, the word S sort of gives you a clue that it scans the surface. Okay. Whereas transmission electron microscope, the word tells you it goes through the sample. So it is sort of cutting or slicing through the sample. It gives you a longitudinal section of that same cilium that you can look from the outside. So this is the surface of the cell which is giving you the cilium. But this one sort of gives you the section of that cilium that you have cut through and through. And you can look at the finer details and observe, make your conclusions. Then we move on to another aspect of cell, wherein it's called cell fractionation. Now cell fractionation basically means you have to take the cells apart from the state of it being just an organ. If you remember your biological hierarchy that you had done, wherein you had, say, the digestive system, okay, of that we picked one organ, say the organ we picked, say, stomach. Now of that you took out tissue of the stomach, which is a collection of cells. Now from that you took out or separated out cells. Now what is inside the cell is what you want to look at. Now so far, now this is like the entire digestive system. This is your stomach for you, and this is just a tissue or a section which has got lots of cells that you have taken out. Okay. Now this, you separate out just the individual cell. Now this cell, you would like to know what is inside it. Just excuse me for a minute. Apologize. I'm just taking time to get used to this particular medium of instruction. So please bear with me. So this is like an individual cell, and the cell organelles is what we are looking at. Now the process of separating out the cell organelles from the cell is called cell fractionation. Okay, you're taking the cells apart and you're separating out the major organelles from one to the other. This uses a particular gizmo called ultracentrifuge. Now this is a centrifuge. Centrifuge basically uses centrifugal force, and it sort of rotates round, kind of like this, and the heavier material sort of settles at the bottom of the test tube, and the lighter material are on the top of the test tube. Now any manual or a method book which gives you the step-by-step instructions in the instruction book will sort of give you the steps that are involved in separating out and obtaining a certain cellular organelle, because people have worked on it, scientists have worked on it from time to time and come up with the best method to separate out and get, say, the ribosomes or the nucleus or the endoplasmic reticulum or the Golgi apparatus or the lysosomes or so on and so forth. So cell fractionation basically enables the scientist to determine the functions of the different organelles that are involved in it. Now we need to understand surface area and the ratio of surface area with respect to the total volume of that material. Now why is this even important? The ratio, if it is very, very high, that means there is more surface for it to react, and there is capability of better reaction that is possible. To understand this, there is many ways to explain this. One of the explanation that I had given my class was you could pack 100 chocolates together in one box and wrap it up with one wrapper, say a wrapper which is, say, 2 feet by 2 feet wrapper. But if you had to take out all those 100 chocolates and chocolate bars and you had to individually wrap them, this 2 feet by 2 feet wrapper would not be enough. You need more. basically, smaller the size, then more surface area there is that is present. And it is making more sense for a cell to be smaller in size. Why? How? We'll go to the next slide where I can do a little better explanation. Now, consider a small piece of, say, agar gel. Okay. Now, this particular agar gel piece is 1 centimeter by 1 centimeter. Okay, it's a small cube. And another one which is 2 centimeter by 2 centimeter, and another one which is 3 centimeter by 3 centimeter. You put them inside, say, hydrochloric acid. Now, and you look at it, because you have already put, mind you, you have put it with some dye or some color around it. Okay, so you can sort of get a feel of the reaction, or exactly how much of the hydrochloric acid has gone in or diffused in. You need to know. So when you have something which is a small piece by 1 centimeter by 1 centimeter, it, after a certain period of time, say 10 minutes. Okay, I would give it 10 minutes, an example. Completely the hydrochloric acid has diffused inside it. Whereas after 10 minutes, only a small amount of it has diffused into the 2 centimeter by 2 centimeter cube. And even smaller amount has entered the 3 centimeter by 3 centimeter cube. Why? Because the rate at which diffusion occurs is faster when there is larger surface area to volume ratio. Basically, this table down here will help you understand what I'm talking about. The first cube, the width is 1 centimeter. The surface area that it has is 6. That is 1 plus 1 plus 1, all the 6 sides put together. The volume that it has is 1. That is 1 into 1 into 1 centimeter cube. Now, if you take the ratio, it is 6 by 1. Surface area by volume. The ratio is 6. Now, if you consider the next guy, 2 centimeter by 2 centimeter, the surface area that it has is 24. The volume that it has is 8. Basically, it is like this. This is, okay, 2 plus 2 plus 2 plus 2, so on. No, this is, I'm sorry, excuse me, 4 plus 4 plus 4. 1 minute. I think I'll just take that out because I don't want my students to get confused. I apologize. Okay. Okay. So this is basically surface area, like 2 centimeter by into 2 centimeter, right? So 4, just a second. So you have it like this. 4 plus 4 plus 4 plus 4 plus 4 plus 4 for all the 6 sides, whereas this was 1 plus 1 plus 1 plus 1 plus 1 plus 1 for 6. Wait a minute, 1, 2, 3, 4, 5, 6. Yeah. Okay. So that is your 6 sides, and this is like the cubic, which is just the 1, and so on and so forth. The way you get it is smaller the size, bigger the ratio, and faster the rate of reaction that can take place. So it makes more sense for a cell. I'm guessing after this you understand this. In short, I will also put another video and on the web links so you can just look at what they're talking about in this, if this particular chart is not clear. I'm guessing by now it should be, but if it is not clear. Okay. Now, the kinds of cells that exist are prokaryotic and eukaryotic. I have already gone over this twice over. Okay. First lab and also first lecture time. and during microscopy I had mentioned this. So you have prokaryotic cells and eukaryotic cells. The difference between the two: prokaryotic means there is no nuclear envelope, meaning the DNA of the bacteria is not covered, whereas in eukaryotic there is a nuclear envelope covering the DNA. Prokaryotic is approximately 100 times smaller than the eukaryote. Eukaryotic, on the other hand, is bigger in size. There are no cellular organelles present except ribosomes in prokaryotes. There are cellular organelles present, and the types of eukaryotes that they are plant cells, animal cells, fungi, and protists. For our requirement, we would be talking briefly about plant cells, animal cells, and bacterial cells a little bit. Okay, this is again just a repetition of the previous page. A bacterial cell, what it contains is the nucleoid. Basically, nucleoid is the nucleus without a cover. Okay, now often my students get confused between cell membrane and nuclear membrane. Cell membrane is outside, covering the entire cell. Okay, nuclear membrane is covering the nucleus of the cell. Okay, so cell membrane exists in all animals. If they didn't, the animal wouldn't exist, or the living creature wouldn't exist. It is the DNA that we are talking about. However, in bacteria, it does not have nuclear membrane. Everything has a cell membrane, please mind you. And I have this mistake often happening, and people misunderstand. My students misunderstand at the bottom line. They just know about a membrane being present or absent, but they forget what kind of membrane we are talking about. The eukaryotic cells, the subject that we are going to deal with is plant cells and animal cells. Okay, we are going to be talking about the different parts of the cell structure, the prominent ones. The plasma membrane, we'll briefly go over the plasma membrane and talk about it in detail in the next chapter, chapter 7. The nucleus is the central guy, the main guy who is responsible for everything that is happening. It's like the Pentagon, okay? Or even I would say the White House, where all the decision takes place. Just outside the nuclear membrane is the nucleus is the nuclear envelope. Okay, it has certain ribosomes present in it. Okay, the way a nucleus is described is you would have, this is the nucleus I'm talking to, this is the entire cell. All right, so this is the nuclear envelope that it has, and then it has certain region which is, now if I'm just sort of cutting it through or giving you a 3D effect, so this is the nuclear pore like openings present on the nuclear envelope, and inside you have like a core of DNA which is very, very tightly bound, and then the outside part of it is loosely bound. Okay, now anything which has DNA plus protein is your chromatin. Okay, then you have the endomembrane system, kind of like once the information, or you pass a law or you pass a certain rule, and what happens to it next after that is what you need to understand. What are the different steps that are involved in the cell trying to fulfill that? Okay, so the nucleus has just got the information, like say it just decides, okay, we have to make X enzyme. So it has the information to make the X enzyme. So let me go to the next slide and show it to you. So this is your entire cell. Okay, this is your entire cell. Now this is your nucleus. I'm just drawing part of it. Okay, completely. Now these are your nuclear pores that are present. The DNA is present inside. Out of the pore comes an mRNA. So the nuclear envelope is responsible for the first step. Through the nuclear envelope comes out the mRNA. Okay, this is the information that has come out. Now we have to read it, translate it, and make some sense out of it, make a product if it's necessary. So things are organized in the cell in such a way that there is a certain sequence of action that can take place. Okay, to remember that, I sort of put together a mnemonic. The sequence in which the action takes place is via the nucleus, which is the initial step. Then you have the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuoles. Okay, these are the sequence in which things occur. So to remember that, I said not every German likes Volkswagen. So if you know this, you know what is the sequence of the endomembrane system and what each thing is responsible for. Not N stands for nuclear envelope. Every, the E stands for endoplasmic reticulum. German, the G stands for Golgi apparatus. Likes, it stands for the lysosome. Volkswagen, it stands for the vacuoles. Okay. We'll go over the different parts of the cell, animal cell and the plant cell, again at the end of the chapter. Now briefly, to just understand, kind of like the diagram I tried to draw a few slides back, this is the central region, which is the nucleolus, which is pretty dense and tight. Okay, this is your nuclear envelope, the purple line. These are your nuclear pores that are present. Okay, these are your chromatin, which have DNA plus some proteins present. All of these together is your nucleus. Okay. Now, immediately after outside the nuclear pore is your endoplasmic reticulum. To understand the endoplasmic reticulum, I want you to think of a croissant. Okay, a croissant basically involves you sort of knead the dough and roll it, fold it up, roll it again, fold it up, roll it again, and you keep doing this, and then you sort of have, like, when you bake it, it is baked in layers, but all of those layers, mind you, are continuous, right? Something very, very similar to that. Around the nucleus, you have a continuous layer, which is like a layer of infoldings of material, which is the endoplasmic reticulum, which completely surrounds the nucleus. This is of two types. One is smooth endoplasmic reticulum, which has no ribosomes and it doesn't look very grainy at all. And then the rough endoplasmic reticulum, which has got ribosomes stuck on it, giving it a grainy appearance. So if I go back to this particular structure here, the endoplasmic reticulum here are all of them, which are the rough ER, and these guys are the smooth ER. Okay. Immediately, now that we have finished not and every, we need to talk about German. So before that, let me just go over with the functions of the endoplasmic reticulum. Okay. So the functions of the endoplasmic reticulum, what they are responsible for. Now, they are the ribosomes that are present are of two parts, as explained. One is a bigger unit and another one is a smaller unit, and this is what is stuck on the endoplasmic reticulum. This is what is giving the grainy appearance that you would see, the dotted kind of grainy appearance which is present. The function is, for smooth endoplasmic reticulum, is lipid synthesis, carbohydrate metabolism, detoxification of poisons, storage of calcium ions. The functions of rough endoplasmic reticulum is to create or synthesize secretory proteins which are transported, and to add membrane proteins. Okay. The Golgi apparatus, on the other hand, okay, is the next in the not every German. The endomembrane system, we finished this, now we are talking about this. Golgi apparatus is responsible for receiving, sorting, shipping and manufacturing. So it also adds finishing touches and refinements in the stages in the different cisternae or vesicles or carrying bags. Okay, and kind of behaves like a post office which receives. So I would like to call the Golgi apparatus similar to a post office. Okay, so the way it works in the next slide, you can see, is from the endoplasmic reticulum, whatever material is being made is sort of given to it. The membrane fuses and passes through, and these vesicles are taken out to the plasma membrane and out they go. Or they could be taken to another region completely. Yeah, wherever that it is required. So it's protected. Then you have the next kind wherein we talked about not every German likes. We are talking about the likes. Likes is lysosomes. Lysosomes contain hydrolytic enzymes. They have a different pH inside them, like a very acidic pH. Very acidic. And it helps for better function of the enzymes which are acting on the material. So at a certain place, that particular pH is concentrated. That does not mean the entire cell has the acidic pH. So it is like a very efficient way for the cell to work with material wherein it needs another kind of pH for better functioning of that enzyme, but it doesn't have to turn the entire cell into that pH level. So what are the different types of things that it can do? It can break down an old organelle. It can ingest a bacteria and break it down. It can ingest any food particle and digest it. Okay, so lysosomes that I have here. Now, an example people have given is a disease called Tay-Sachs syndrome, which affects certain races. In this case, most often seen in Ashkenazic Jews from Eastern Europe. This particular disease is very prevalent in their community. Now, not so now, but it was sort of very prominently observed as a disorder that would occur, genetic disorder that would be commonly occurring in that particular race. They contain defective hydrolytic enzymes, so they cannot digest the lipids. So that means there is accumulation of lipids in the brain cells, which leads to an impaired brain for people who are affected by this syndrome. So this just goes to show how important lysosomes are. Okay, so now this picture I tried to draw and put together what you could do, like a white blood corpuscle or a WBC or a macrophage, that's what it's called. They have a lot of lysosomes in them. Why? What? Now imagine this, a macrophage. The orange circles are your lysosomes. There is a bacteria. It sort of ingests the bacteria. The process of ingestion like this is called phagocytosis. Okay, it ingests and takes it in. It is inside a vesicle. All of these guys come together. The membrane between the two dissolves and it becomes a bigger circle with all the enzymes being released to break down the bacteria and digest it completely. Okay. This is another picture which could give you an idea of what is phagocytosis means. Phagocytosis is just ingesting a bigger particle for the purpose of digesting it. Autophagy basically means ingestion of cells' own cell's own material, kind of like a mitochondrion which is old and sort of gone under disrepair. Instead of repairing it, they sort of digest it out and recycle the components. Okay, now vacuoles. This is the last of the endomembrane system. We so far covered in the endomembrane system. Not every German likes. Now the Volkswagen part is what I'm trying to cover. Vacuoles. Now this is the last of it. They have greater significance in plants. They are present but in smaller size in animal cells. Present in smaller size in animal cells. Now, okay, I want to give you an example. Supposing you have a vessel and you fill it with water, say 1 gallon. Here. Okay? You fill it with water. Now, in that same vessel, you place a bigger big block of a vessel inside it. Okay, so this is the block you have put inside. And now fill it with water. And check the amount of water that is required to fill it. It will definitely be less. Why? Because this block is occupying a great surface area, a greater area or a greater volume, and so whatever the cell needs is only what area is left over around it. So if the cell, an example for us in this case, a plant cell has a big huge vacuole present in it, it does not have to invest in a lot of energy and food material and whatever is resources are available to increase the content, cytoplasmic content. Instead, all it can do is continue with its whatever metabolic activities it does, and the vacuole size sort of does not impose upon it to create more cytoplasm. Okay, so that is one very important thing, and it is like an energy investment situation, energy saving situation. Okay, what are the functions of the vacuoles? It is similar to a waste bin. It can contain pigments which give color to flowers. It can contain bitter products which are dumped into it to help avoid being eaten by herbivores. Okay. Now, you also have another kind of vacuole. This vacuole is called the contractile vacuole. We talked about what a central vacuole and how it will be helpful, what a food vacuole and how it can, in conjunction with a lysosome, sort of help digest. Now, the contractile vacuole is seen very clearly in an organism. It is a protist, okay, and it's called paramecium. What it does is it is responsible for taking out excess water. So it sort of maintains the particular cell with a certain amount of water and nothing more than that. Okay. It also is a repository, or meaning storage for potassium ions and chloride ions. Okay. This is your central vacuole for you. We are finished with the endomembrane system. Then we move on to talk about the different special organelles that are present in a eukaryotic cell. Mind you, all of this that I'm talking to you is present in eukaryotic cells. So there are certain things that are common in all and commonly present in all eukaryotic cells. So if I'm talking about plant cell and animal cell, so far whatever I spoke about, they are generally present in both plant and animal cells. Now take the example of mitochondria. This is a special organelle, and this special organelle is also, please remember, is present in both plant cells, animal cells, and in fungi and in protists. Mitochondria has to be present in all eukaryotes because it is the site for cellular respiration. Okay, it has got a double membrane. The reason they are talking about a double membrane is it goes back to a theory called the endosymbiont theory. Now this endosymbiont theory basically deals with a situation or proposes that long ago, when life had just started appearing, wherein the conditions were pretty anoxic and the organisms had to use something other than oxygen in order for them to derive energy. And at that particular point, maybe there was a mutation at some point, and there was one kind of organism which could use oxygen as its source, wherein in the end product of respiration, it was capable of bringing, or it was capable of tapping more energy. Now this organism probably needed, say, protection, in which case another organism sort of said, okay, you exist inside me in a symbiotic relationship. Kind of like, remember the cows and the organisms which can break down cellulose, okay, or termites and organisms which can break down cellulose. They exist in symbiotic relationship. Okay, similar to that, they proposed that a mitochondria might have been an individually replicating organism which somehow got inside another larger organism and stayed, and that is the reason why it has a double membrane. Okay, now this goes on. This theory is also equally valid for a chloroplast, which we will be talking about in some while, in a few slides from now. Okay, the structure of mitochondria looks like this. The simplest, it is present in all eukaryotes. It is autonomously replicating, meaning it can replicate on its own. And if you need to understand more about the endosymbiont theory, you can go to page 109 in the textbook. The way it looks is the outer membrane. This is the outer membrane. This is the inner membrane. And this is the intermembrane space. This is the mitochondrial matrix. The infoldings of the inner membrane is called cristae. Excuse me. I apologize. Then you have the next organelle, special organelle that we would talk about, and we would be talking more in detail about it when we are doing the chapter on photosynthesis. It is again capable of having double membrane. It is the site for photosynthesis, so it is present only in plants. Of the eukaryotes that you would see in plants, it is present. Okay, it also has an outer membrane and an inner membrane. The structure, the way I would like to put it across, is this. You have your outer membrane, you have your inner membrane, you have the liquid that is present. It's called the stroma. And then there are these stacks, okay, called like stacks of coins or stacks of poker chips. Stacks of coins, one above the other. This is called the granum. Okay. Now each coin is a thylakoid. Inside the thylakoid is the lumen or a liquid present. The membrane of the thylakoid is the most important thing, which contains the photocenters, which is capable of photosynthesis. Okay. Synthetic pigments are present in photocenters. Okay. Many granum together are called grana in plural. Okay. Then you come across another organelle called peroxisomes. Now peroxisomes remove the hydrogen from fatty acids and break down fatty acids into smaller molecules. This is added to oxygen to give you hydrogen peroxide. Now this hydrogen peroxide is poisonous, and this can cause a lot of damage to cell. So the peroxisomes have another enzyme called peroxidase, which can break it down and release water. Okay, so peroxisomes have enzymes to remove hydrogen from fatty acids. So they are responsible for breaking down fatty acids essentially. So they have these enzymes and they have a backup peroxidase to take care of the poisonous hydrogen peroxide that can be released. Okay. In plants, especially in the seeds, peroxisomes are called glyoxysomes, and they are in high content, and they are present or observed prior to the leaf formation in a germinating seed, because till then the plant needs energy, and the energy cannot be got by photosynthesis because it doesn't yet have leaves. Okay, that's what I have written. I have rewritten exactly what I was talking about. So this is your peroxisome here. Okay, then we go on to the part of the cell, which is, I would like to call it the packing material. Imagine if you have to send, say, a glass vase to someone, flower vase to someone, okay? And you would put it inside a box, and then you would sort of stuff it with some material in the sides, like an airbag, like how any packing material comes, or these small bubble wrap or anything else. So that sort of keeps everything in place, and the vase doesn't move, the vase doesn't break. So how can everything stay inside your cell in the same position? For this particular purpose, you have the next organelle or material that you're going to be talking about, which is the cytoskeleton. Okay, this is responsible. Main functions is support and movement. It is of 3 types. Now there is so much material covered in the textbook for cytoskeleton. I've sort of simplified it. Okay, microtubules, microfilaments, intermediate filaments. Microtubules are made of tubulin protein. Microfilaments are made of actin protein. Intermediate filaments are made of keratin protein. Intermediate filaments are more permanent, whereas microtubules and microfilaments are more temporary. Okay? Now, the important microtubule play an important role in cell division. They grow out from an organelle called centrosome. Okay, in an animal cell, the centrosome has 2 centrioles. Now, centrioles are absent in plant cell. Okay, this is kind of like a structure that you can put up here, then dismantle it, and then put it up another place, kind of like a tent you have. So you have all these bamboo, the sheets, the support system and everything, and then you can dismantle it, take it somewhere else and put it up there, and then you can dismantle it, and so on and so forth. This sort of explains that they are temporary. Okay, now another thing that you would continue to understand in this section is microtubules are also present in cilia and flagella, made of the tubulin protein. Okay, it is mostly flagella, that is mostly seen in animal cells. Cilia is shorter, flagella is longer. Cilia moves the material over the cell, flagella moves the entire cell in a certain direction. An example of cilia is the cells present in lungs called the alveoli cells have a mat of cilia over them, which sort of move in a direction and move the material which is on top, kind of like any particle that they need to cough out. The other example in flagella is like a sperm cell. Okay. So that is now defective cilia, okay, or defective flagella in humans can lead to infertility, males especially. And cigarette smoke products paralyze the cilia. This is a known factor. Now, this picture, normally if I take this lecture in class, I just bring the laundry softener sheets, laundry dryer sheets, and you can see how fibrous it looks. Something similar to that is what this looks like. So I just used to bring that normally as my analogy. Now, the centrosome from which arise the two centrioles are like bumpers. Like if you have like a train, now this is the station and it has certain bumpers here, and this is your train which has come. It will have certain bumpers. Okay, so these bumpers sort of hold the train back and not let it crush the wall. Similar to that, these centrioles do not let anything, any pressure crush the cell. Okay. This again, I just wanted to show flagella would move the entire cell, whereas cilia, the movement would sort of decide what direction the particles would move. Kind of like when you're rowing. Okay. Then we move to the last part of this particular cell, which is called extracellular matrix. Okay, it's called extracellular matrix. Now, extracellular matrix is very commonly seen in animal cells. It is made of collagen. Okay, and I would like you to think grout, kind of between two tiles. If I need to join them up, I would put grout, right? So extracellular matrix is something similar to that. There are certain membrane proteins that can interact with it and hold it in place. In plant cells, the cell wall is outside the cell. It is rigid, cage-like, and it is made of three parts. The primary cell wall is thin. The middle lamella is grout-like, similar to the extracellular matrix. It is made of something known as pectin, which is also used in jams and jellies to thicken it. Okay, and then it has secondary cell wall, which is perforated. Then you move on to certain forms of cellular communication. Okay, these in animal cells, again, you have three types of cell junctions. One is the tight junction, which is like nailed together. Then another is desmosomes, which is like rivet. For this, I would like you to think IKEA furniture. Kind of like if you have a piece of material, then you have a riveting piece which sort of holds it together. If you have put together any furniture like that, so this is like present in between and it holds the two planks together, okay, in between the. And then you have something known as gap junctions, which is like a channel, communicating channel between cells. On the other hand, plant cells have only one kind of cell junction, which is channel-like, and it's called plasmodesmata. So this is your cell, plant cells, which have these connecting channels between two cells, and they are plasmodesmata. Okay. So the tight junctions prevent leakage. Desmosomes are anchoring junctions. Gap junctions are cytoplasmic channels. Kind of what this looks like. A tight junction sort of prevents fluid from moving across the layers. Okay. And in desmosome, it sort of rivets the cells together. Gap junctions are channels through which the material can move from one cell to the other. This sort of completes this chapter, and this is like a précis of all the organelles, just showing you what the structure looks like, what is the structure and description, and what is the function. Okay. And I hope you are able to go over this chapter on your own sincerely, with the textbook, with a paper and a pencil to take notes and understand it. Wherever you're stuck, you mark that and we can discuss it in class when we next meet. This is extremely essential because I am not going to teach this chapter one more time because I've put it online. We will definitely, however, work on a worksheet which will be scored, and I can talk to you about it in detail next time. So thank you very much, and hope everything was understandable. Hello.